Hello, my name is Uyen Bui and I'm an undergrad student in the POKY team. Today I will be giving a tutorial about strip plots. So here are a couple of things you can use strip plots for and why it's needed. The first couple thing is sequence backbone assignments. You can use this to primarily walk up along the protein sequence. You can do this by extracting the 3D NMR data and then lining them up and then matching the chemical shifts of the residues. Another one is if you have a lot of congested 3D data, this happens a lot with large proteins where it creates a crowded spectra, and you can use strip plot to isolate a single peak to show the specific signal that you want over that one amino acid. Then you can also do this with inter-experiment comparison. You can do this by overlaying the strips from different 3D experiment spectra to help distinguish between the intra-residue peak and the inter-residue peak by seeing which one disappears in your selective experiment. Aside from backbone, you can also do this with side chains with the 3D data from TOSY or NOSY experiments to identify the protons or carbons belonging to the specific side chain to determine their full 3D structure. Lastly, you can also use this for quality control to scan the completeness of your own data and to ensure that there's no missing peaks or gaps in your strip plot sequence. So here are a couple of the things that we can use strip plots for. I will now be going on to POKY itself and showing you how to use strip plots. Here I am now on POKY and I have the three spectras I will be using. I will be using HNCACB, N-HSQC, and CBCACONH as my spectras. So as I move on to POKY, you can already see that I have my spectras open, like I said. After doing automatic peak picking with APES or iPick, or getting assignments for iPine, the best way to validate your peaks or your assignments is using strip plots. So first, you want to open strip plots using two-letter code SP, and the screen will pop up. As you can see, then you will go to Show. Then you will go to Select Strip Spectra on the first one you see, and it will show you the two spectras that you want to put into your strip plot. I'll be using CBCACONH and HNCACB for my examples. On the little box over here, you want to click it to ensure that it is checked. And then you can also show the spectrum name under each strip to avoid any confusion. Click Apply, OK. Then I will be going back to my N-HSQC, zooming into whatever peak that I want to validate. I'm going to select the peaks, the peak that I want. Then go back to add peak right here. And then it would add the peak from both the CBC, CONH, and HNCCB. Now we'll go through a couple field ones to show you the field pairs. Again, I will go to this one down here, click on it, add peak, and then here, as you can see again, it has added the two spectras, and it has the names a little bit down here so that you do not get confused. Now do this once more with this peak right here. Click it, add peak, and it has shown both the CBC, CONH, and HNCCB. And that is how you use strip plots.